All right, I'm going to go ahead and get started. I'm sure we'll have a couple people popping in. I did get a lot of emails from students talking about power outages and still lacking access to Wi-Fi. So I will be recording this and putting this on the YouTube channel as per usual. All right, so hopefully you guys fared the storm all right. No damage to your house or your cars. I posted these two videos, the links for you guys for 6.1 and 6.2. 6.1 is an introduction to DNA, talks about the structure, the importance or significance of DNA. And then 6.2 talks about replication. And I cannot emphasize the importance of watching these videos enough, especially when it comes to replication. Because if you don't understand these first two videos, when we get to transcription and translation, you're going to be in some trouble or up a creek without a paddle, swiftly and quickly approaching a waterfall. All right, so we got week 11, and at first I didn't think we would be out today, but that's all right. That's how life goes. So I went ahead and I'm postponing all the due dates, especially for the quiz. Wait for this to load. So I'm going to move the quiz till Monday, right? And so I'm going to review a little bit today. And hopefully you've had a chance to get the vocab in, and hopefully you'll have a chance to complete your notes. But obviously, because of some individuals not having internet, I'm going to really wait till Monday before I start looking at those things. So our quiz will be on Monday, and our quiz on Monday will cover the first 12 or 13 slides of the PowerPoint. And I'm going to review a little bit today, and then I'll also do a quiz view on Monday for you guys. All right, so it's going to cover DNA structure, the function of DNA, as well as DNA replication. And so I'm going to go ahead and pull up the PowerPoint and just kind of go through a little bit of a highlight, right? I know that hopefully you guys have all watched the videos, but if not, right, this will still give you a good introduction. And if you already have, this is a nice little review. Even if you haven't watched the videos, I strongly suggest it because I don't have time to go over everything today. So with DNA and RNA, okay, we know DNA is a nucleic acid. It's present there in the name. And the deoxyribo part is talking about the sugar, okay? So DNA is a nucleic acid. All nucleic acids are made of nucleotides. And every nucleotide has a sugar, a phosphate, and a nitrogen base. So the sugar that's present in DNA is deoxyribose, and that just simply means that an oxygen had been removed off from one of the carbons. Now, what is the function of DNA? DNA is the universal genetic code, so all living things have DNA. It's what we talked about in the very, very beginning with the 8 characteristics of life. But why is DNA so important? Well, DNA contains our genes, and our genes are like recipes or instructions on how to make certain proteins. And we're walking, talking stacks of proteins. And so the proteins that are essential for our life's functions are all encoded for by DNA. And so, when our cells need new proteins, they can consult with or go read the gene or chapter in DNA for that particular protein, and they'll know how to make it. And DNA acts as a molecule of inheritance or hereditary, right? So it's passed from one cell to the next. So that ensures that, the new cells have the instructions they need. And if we're talking about sexual reproduction, we're making sure that the new individual has the instructions they need to survive. Now, as I had mentioned, DNA is made of nucleotides. Those are the building blocks. They've got a sugar, phosphate, and a nitrogenous base. And if we look at the sugar carefully, the deoxyribose, you can see that an oxygen has been removed right here. So normally if you were to put an oxygen back, this would be ribose. And so since we remove that oxygen, it's deoxyribose. One thing I want to point out are these numbers are referring to carbons, and they're just a point of reference. 1, 2, 3, 4, 5. The 1 prime, 2 prime, 3 prime, 4 prime, and 5 prime, the little dash we call prime symbol. And that just tells us which carbon we're referring to. So I would say that the 2 prime carbon on deoxyribose had an oxygen removed. And these numbers are important when we talk about DNA replication. Now, not all DNA nucleotides are the same. There are 4 different nitrogen bases that can be found in the nucleotides of DNA. And so therefore we have 4 different nucleotides, and the differences would be the nitrogen bases. Because you look at all 4 of these nucleotides, they have the same sugar, deoxyribose, they have the same phosphate group, but the nitrogen bases are different. We've got adenine, thymine, guanine, and cytosine. You can see that there's similarities between some of them, and then there are differences, right? So adenine and guanine are what we refer to as purines, right? They have 2 rings in the nitrogen base, whereas pyrimidines, such as cytosine and thymine, they only have a single ring. And I remember this by see the pyramids. You need to make sure to see the pyramids during your life. And you can see that this is a DNA strand, maybe a single one. And we've got 1, 2, 3, 4 nucleotides held together in this strand. And you can see that although adenine and guanine are very similar in the fact that they've got 2 rings, you can see some differences, right? You can see this double bound oxygen here that's not present over here. And you can see the same differences when you look at cytosine and thymine. So although they're very similar in structure, these slight chemical differences will change their behaviors. Now, the overall structure of DNA is actually made of 2 strands, right? And each strand will be made of many nucleotides, and these 2 strands are held together, and they form what is known as a double helix, right? So DNA has a double helix, double meaning 2 strands, and then helix referring to that kind of spiral staircase shape, okay? So it's a double helix. And you can see that there are some significant components, right? We've got these strands. These would be the sugar phosphate backbones. And then we can see the latter rungs of the inside. Those would be our base pairs, right? Our nitrogen bases that are held together. We won't worry too much about the minor and major groups, but these numbers, these 3' and 5' will become rather important. So if we take our double helix and we kind of iron it out so we can better study the structure, we can see that there's sugar phosphates along the backbones and nitrogen bases running up along as ladder rungs. And we can look even a little bit more closer. We can see we can identify an individual nucleotide, that'd be deoxyribose, phosphate, and in this case would be cytosine. And you can see that these sugar phosphate backbones are just repeating, right? So there's no variation here, whether we're talking about this side or that side. It's just sugar, phosphate, sugar, phosphate. The real difference, or the real code, or the real power of DNA is actually in the order or sequence of these nitrogen bases. Another thing you might notice is that certain nitrogen bases only pair with certain nitrogenous bases. You can see that C likes to pair with G. I can see evidence of that here as well as down here. And I can see A and T wanting to pair together. And they're held together chemically by a hydrogen bond. And you can see that C and G have 3 and A and T have 2. And remember, from the very, very beginning, hydrogen bonds are not very strong bonds. And so the weak or the nature of these weaker bonds become very important when we talk about DNA replication. Whereas the sugar phosphate backbone, the backbone of our molecule is held together by stronger covalent bonds. And that's a good thing because you don't want the backbone of your molecule falling apart. Another thing you'll probably quickly notice is that the sugar phosphate backbones aren't necessarily exactly the same, right? They're kind of inverted, right? One's going in this direction and one looks to be upside down. And so we refer to that as being anti-parallel. Anti or anti simply means the opposite. And parallel means that they run alongside of each other. So they run alongside of each other, but kind of in opposite directions. The lanes of traffic in a 2-lane road are anti-parallel, okay? So they're parallel, they shouldn't really cross, and they are moving in opposite directions, anti-parallel. And so what this means, or why this is important, is we can refer to the directionality of these DNA strands using that 1', 2', 3', 4', 5' carbon. So this would be 1, 2, 3, 4, 5. And so this right here is the 5' end of this particular DNA molecule. And then down here, we've got 1, 2, 3. This would be the 3' end. And since DNA is anti-parallel, and since this strand is kind of upside down, it's the opposite. This would be 3', and this would be 5'. Now, let me give you a really easy way that I've used over the last decade to remember this is, I think we always jokingly call this a swimming pool, right? And if I think about water parks and some public pools, you have to pay money to swim in the pool. So I think $5 to swim in the pool. So what I do is when I look at these strands, I'm like, all right, where's the swimming pool at? All right, there's the pool. That's 5'. I look down here, I'm like, all right, that's the end that has the pool. So that's 5. Another way you can think about it, it's like real estate, right? There's no pool over here, so it will be worth less than the house with the pool, right? I don't know. It worked for me. You just need to know that there's a 5' and 3', and since DNA is anti-parallel, the opposite side would be different or inverted, if you will. Oh, let's clear the drawings. And the nitrogen bases follow what is known as a complementary base pairing rule. So we've got adenine, thymine, cytosine, and guanine. And I think straight line with straight line, curvy line with curvy line. So when you draw a capital A and a capital T, you use straight lines, whereas cytosine and guanine or C and G, it's a curvy line. So that helps me remember who pairs with who. Some people also say apples in the trees, cars in the garage, whatever you need to remember it. Another thing to notice is the number of hydrogen bonds. So adenine and thymine only have 2 hydrogen bonds, while cytosine and guanine have 3. And so who likes who more? That would be cytosine and guanine. So areas where you find lots of cytosines and guanines, the DNA is a little stickier, whereas the A's and T's would be places where there'd be less energy required to break those hydrogen bonds. And in the video on 6.1, I talk about the importance of pairing purines and pyrimidines together. And so you'll want to make sure to watch that to get those details. Now, here is the real intense portion of the discussion, right? It's DNA replication. So buckle your seatbelts. It's kind of wild. And even if you watch this and you hear me talking about this, I still recommend you go watch the video. So DNA replication is a process through which our cells make copies of our DNA. When does it happen? So it happens during interphase, and not just any old part of interphase. It happens during the S phase of interphase. So if we're talking about any kind of cell, we're first going to have interphase. We're going to have a G1 period, which is a period of growth and development. And then the S phase is when the cell would be copying all of its DNA in preparation for division. That would be followed by G2, a final stage of preparation, and then the cell would divide. Maybe it's binary fission, maybe it's mitosis, maybe it's meiosis. I don't care what kind of cell division we're talking about. We need to make sure to make copies of DNA before the cells divide, because the DNA is an essential instruction manual. It contains all of our genes, and if we're going to take one cell and we're going to make it into 2, if we're talking about binary fission or mitosis, we have to make sure those new cells have the correct amount of DNA. They have to have identical copies of instructions that we started with. And in the case of meiosis, obviously we're dividing not once but twice. We have to make sure that each of the gametes at least has half of the set of instructions necessary. And then replication does use that complementary base pairing. And so when we use our original strand and trying to make copies, we use it as a template. We look at the code and we try to pair it up with the complementary base pairs. And then DNA is semi-conservative, which means we start with one original molecule, or sometimes people call it the parent strands of DNA. And through DNA replication, we take the original double helix and we split it in half. And as we split it in half, we kind of read the letters and play by the complementary base pairing rules, and we make a new strand. And so eventually this whole blue original parent strand will be unzipped and it'll be copied. And what we end up with are 2 identical molecules of DNA that both contain one strand from the original and one new strand. So that's why they are semi-conservative. Semi means half and conservative means saved. And so we would call these replicas of the original. So if I have a World War I replica rifle, that doesn't mean that it's an exact, it wasn't there in World War I. It's a copy of the original, right? And so these would be replicas of the original. They're identical, right? For all intents and purposes, these DNA molecules are the same as one another as well as the original, because we want both new cells to have the same set of instructions. Now, who gets this done? Well, obviously it's going to be enzymes, right? Because we're talking about building and breaking and separating and putting back together. And we know that enzymes are going to do this. Enzymes are going to catalyze all these reactions. And there's actually one enzyme I do want to include here, topoisomerase. Okay. So helicase is an enzyme that unzips the helix, hence the name helicase. Primase is like a painter putting on a layer of primer paint. If you're going to paint a car or paint a wall, you need to lay down like a little bit of a starting point, right? Or something to start with or something to paint on top of. That's what primase does. DNA polymerase is the real workhorse here. DNA polymerase builds or synthesizes the new DNA polymers, right? So it's going to be responsible for reading the original code and synthesizing the new complementary strands. Ligase is kind of the lick it and stick it. It's going to take the fragments at the very end and lick them and stick them together and make sure that they're all good, right? Ligase is going to make sure that everything is done properly and make sure everything looks good and is glued together before the DNA can be utilized or separated into 2 new daughter cells. And then telomerase is a really neat one, right? And I'll talk about this separately later on. And then topoisomerase just kind of holds everything still while we're working. So let's go ahead and get started. Clear this. So I'm going to draw this. And if I were you guys at home, if you have a piece of paper or pencil readily available, I'd go ahead and grab it, because I really think the best way to learn this is by drawing it and writing it out repeatedly. All right, and I am going to explain it a little bit different than I did in the video, so that way you've got two different kind of versions to refer to. But I would definitely recommend getting a piece of paper. So here we have our little cell. And inside of our cell, we have the nucleus. And inside of the nucleus, we have our chromatin, right? And then we enter interphase into the S phase of interphase. We need to make copies of our DNA molecule, right? And so we know that DNA normally, right, during normal situations is kind of loosely coiled as chromatin and would have this double helix shape. Well, during replication, the double helices and double helix would need to be unzipped first, right? And so when we unzip the DNA molecule, what we end up with are two little naked strands of DNA. Now, the first question is, how is this being done? Well, let me change colors. There is one enzyme upstream that's kind of holding everything still, right? And that is topoisomerase. This enzyme is just kind of holding everything still while we are, separating those hydrogen bonds and then adding the new nucleotides. It's kind of like if you were to cut a board with a saw, you might use some clamps to hold everything still, or you might use your hand, or you might use, some jigs to hold the wood still while you cut it, or even just using your hands to cut fabric or paper with scissors. The topoisomerase holds it still while you make the cuts and while you work on the material. So that's topoisomerase, just kind of holding everything still and preventing supercoiling from, you don't want it to fold back over on itself. Okay, and the next enzyme, right, is going to be the enzyme responsible for breaking down the hydrogen bonds, right, kind of unzipping the molecule, and that is going to be helicase, right? So helicase, right, this comes from helices, which would mean multiple helixes, and since it's an enzyme that deals with them, we add ASE. So this little helicase enzyme is going to slowly unzip all of those hydrogen bonds, separating the DNA double helix into two naked little strands that are open. Now, what happens next? Well, all along our DNA molecule, we would have exposed, and I'm just kind of randomly throwing some letters down. Okay, we'd have exposed nucleotides, and there are little proteins that attach to make sure they don't fold back over on themselves as well, and kind of holding everything still. And, I know that this is one strand, and I know that this other strand over here would be complementary. It would have the complementary base pair. So what would be on this side? Well, it's complement to A, that'd be T. Complement to T would be A. And then I keep looking at G, C. And what I'm doing here to figure this out is I'm just looking at who would be the corresponding base pair. And, who does G like to pair with? That's C. Curvy line with curvy line, straight line with straight line. Who pairs with A? That'd be thymine, adenine, thymine, cytosine, and cytosine. Whew. All right, now we're good. And you can see that these would glue together if they were able to be close enough to reattach. And another thing we need to think about is the directionality. Remember, DNA is anti-parallel, so they're going to have some directionality. Let's just say that on this strand, we've got the little phosphate sticking out. And oh, hey, it's a pool, right? Oh, yeah, $5 to swim in the pool. So that would be the 5' end. And if this is the 5' end, I know that this end over here would be the 3' end. And since DNA is anti-parallel, I know that if this end is 5', that means that this end would therefore be 3. And over here, this section right here would be 5'. Because anti-parallel means they're parallel, but they run in opposite directions. And that's important for these next couple steps. All right, what happens next? Well, we have to get ready to start synthesizing a new strand. And just like getting ready to paint a car, or getting ready to paint a house or a wall, we need to lay down a primer. So we have these enzymes. I probably just made a bunch of, okay. We have a bunch of enzymes known as primase that will come and attach, and they'll give us a starting point, right? And so this right here would be a primer. Oh, And primers are laid down by primase, right? And it's really kind of weird. They're actually made of RNA, but it gives us a starting place. It gives us a place for DNA polymerase to attach, and kind of tells DNA polymerase where to begin. All right. Oh, shoot. I'm sorry. My mistake. We got one thing we need to correct. Oh, no. Sorry, y'all. I got a little ahead of myself and actually switched. Wrote some things. This would be 3', and then this would be 5', just because of where I put the primer. You can easily fix it in your drawing. Okay. So I accidentally deleted the letter. It's okay, easy to replace. All right, here we go. Sorry about that. We usually read DNA from the 3' to 5' direction, which makes sense. It's kind of like reading numbers. 1, 2, 3, 4, 5. So we always start on the 3' end and we work towards the 5' end. So we're going to lay a primer here, and then we're going to lay a primer over here, another starting location, right? So those are both primers. It just says, hey, start here. And then, let me switch colors. Let me use green. A larger molecule will attach, and a larger enzyme. This is DNA polymerase. So DNA polymerase will find the primer, and then now it knows where to start. And DNA polymerase will read the nucleotides, it'll read the code, and it'll read it from 3 to the 5' end. And as it reads, it looks, it goes, okay, G, who does it pair with? C. A will pair with T. T will pair with A. T. G, C, G, A, and T, right? And so the DNA polymerase enzyme lays down the complementary strand of DNA. And this particular DNA polymerase is reading from the 3 to 5' end. So it'll eventually end up right here behind helicase, just kind of sprinting up behind it. Now on the opposite side, things get a little weird. Right, our DNA polymerase enzyme will attach near the primer so it knows where to start, and it's going to read the strand 3 to 5, right? So it's got to start here and it's got to move in this direction. And as it does this, it reads each one. Okay, T will pair with A, T, C, G, C, A, T, A, and then eventually G. Okay. Now here's the weird thing. This particular DNA polymerase is working towards the helicase, like towards what we refer to as the replication fork. Whereas this particular DNA polymerase is actually synthesizing a new strand of DNA away from the replication fork. So eventually, as helicase continues to unzip those hydrogen bonds, this particular DNA polymerase is going to be right behind helicase. However, this DNA polymerase is kind of working in the opposite direction. And then eventually there'll be an exposed strand or piece of DNA where we'll have to lay down a new primer. And then another DNA polymerase enzyme will need to attach and synthesize the new strand of DNA. So since this DNA polymerase just gets to follow helicase the whole time without ever stopping, we call this side the leading strand. And this would be a form of continuous replication because it doesn't stop. It just keeps going and going and going until the helicase enzyme unzips the whole thing and until this DNA polymerase reads it and synthesizes a new strand all the way to the end. Whereas on this side, the DNA polymerase attaches the primer, synthesizes a new strand, and by the time the DNA polymerase gets over here to the end, there's a whole new exposed section of DNA upstream, and it might have to detach and then sprint back up the helicase, reattach, and then synthesizing another strand. And so it does it in fragments. And so we call this side the lagging strand because it's going to be working a lot harder and it might be lagging behind. And those fragments of DNA that are formed are called Okazaki fragments. I know it's kind of a neat name. It's named after the scientists that discovered them. And while it synthesizes these fragments, towards the end, it needs to make sure these fragments get stuck or glued together, and that's where ligase comes in. So ligase is the enzyme that comes in and glues the Okazaki fragments together, replaces the RNA primers, and makes sure that we have two new beautiful molecules of DNA, both of which that contain double strands of DNA, one from the original and one newly synthesized strain. And you might be thinking, well, where is telomerase? And I'll get there in just a second. So I'll save that drawing just in case. Now here's a diagram, and there's a reason I draw it first before I show you a diagram, because I mean, the first time you see a diagram like this, it can kind of give you an anxiety attack. So here we have our double helix, we have our topoisomerase holding it together, holding it still, while the helicase enzyme breaks those hydrogen bonds. We end up with 2 strands of template DNA. And then you can see that because of the orientation, right, being anti-parallel, we've got, one DNA polymerase enzyme that may have attached up here at a primer and it's working in this direction. That would be our leading strand, because it never has to stop. It's just going to kind of run right behind helicase. But on this side, things are getting kind of weird, right? Because of the orientation of the DNA molecule. We're going to have to lay lots of RNA primers. We're going to have to put down a lot of primers using primase. And then we're going to have to synthesize these new strands of DNA in fragments, those Okazaki fragments. And when these Okazaki fragments need to be repaired or fixed or glued together, that's where ligase will come in. It'll kind of attach itself and glue these little pieces together. And so that would be the lagging strand. Now, telomerase is a wild enzyme that deals with the telomeres. Now, all strands of DNA, all chromosomes, have these little protective caps, and we call them telomeres. And these protect the important pieces of DNA in our chromosomes. Now, telomeres are actually made up of repeated junk sequences of DNA. It's just nonsense that's tacked onto the ends of DNA. And that's because when DNA is replicated, it's not fully replicated. Because of that weird 3', 5' thing, each time DNA is replicated, we end up with an overhang that doesn't get fully copied, and that can slowly, over time, shorten the DNA molecule. And these telomere ends buy us time. Some scientists even think that telomere ends are what determine the lifespan of an organism, right? So the telomeres, every time our cells divide, they've got to replicate DNA first. And every time we replicate DNA, the telomeres get shorter and shorter. And once we run out of this telomere sequence, the important genes, or the coding portions of our DNA, start to get broken down and start to get lost through replications. And so the cells don't function anymore, right? And this is called senescence. The cells just simply stop dividing. Now, in some cells, it's important that they never stop dividing. And so they would have an active enzyme known as telomerase. And telomerase goes back and repairs these telomeres. It continually adds junk DNA to the end so that these ends don't slowly shorten over time. Some examples of cells like that would be, embryonic stem cells, because they got to divide so much in the beginning to develop the larger embryo that eventually specialize into the fetus. Even some adult stem cells have active telomerase enzymes. And then the cells of the gonad, right? The germ cells that produce gametes, right? So in male mammals, most male mammals can produce viable sperm over the course of their entire life, right? And so those individual spermatocytes, right, those germ cells that make sperm, they're going to undergo meiosis over and over and over and over again. And as you replicate DNA, you don't want to be shortening that, because that would mean that potentially a sperm would not be viable because they'd have shortened chunks of DNA. And so they would have active telomerase enzymes in the germ cells repairing those telomeres and making sure that they stay the same length. Now, sometimes in cancer cells can sometimes have activated telomerase genes that produce telomerase, and this enzyme will continue to protect that telomere and allow the cancer cell to divide and divide and to divide and divide. So if we can come up with different forms of chemotherapy or some kinds of targeted therapy that could disable telomerase or turn off the telomerase gene, then we could potentially push cancer cells into senescence, where they would stop dividing on their own. Now, I know that's a lot to take in, and don't worry, telomeres and telomerase are not really going to be on your quizzes or your exams in my classes. It's just something I wanted to throw out there so we can kind of get a better understanding of how cancer could work, how cancer functions, and as well as kind of get a glimpse into how cells, these individual living components, kind of determine and control literally everything, even down to how long we live. Does anyone have any questions up to this point? I know I've been kind of going a mile a minute, but I think there's a chat. Nope, someone's name. Does anyone have any questions? What happens when we make a mistake? That's called a mutation. Okay, and you need to watch the video on mutation that I posted, 6.3, I believe. I'll send out a link to that here shortly. Does anyone have any questions about the structure of DNA or replication? I know this is complicated. What I would really recommend doing is going on Google and Google simple DNA replication diagrams. And then you could also go onto Google and look up DNA replication videos in addition to the one that I produced. And you just really got to look at this, multiple times. I mean, let's go do it really quick together. If I type in DNA replication simple diagram, I can look. And this is, I think, helpful. We can look at different diagrams and be like, okay, what's going on here? Okay, so this is really simple, right? This is just showing us the difference between the leading and lagging strand. Right? You can see that continuous and discontinuous. And so obviously ligase is going to be really important here. And then we look at a diagram like this, very similar to the one that I showed you, right? You can see, topoisomerase and helicase and the leading and lagging strands. Some are a little bit more cartoonish, but they might help. I like this one. This is a pretty good one. I used to use this one back in the day, right? You can see the chromosome, the DNA, helicase working, and the color coded, making sure that they're complementary. And what's really neat about this is when this is done, both of these DNA double helices are identical. They're exactly the same as the other. Now, as far as YouTube references are concerned, there are 3 channels that I highly suggest for studying biology. Khan Academy is a good one, right? And so, I slotted the spelling of that. Right, you just simply type in Khan Academy DNA replication. Khan Academy tends to be a little higher level. It's kind of like probably more on the verge of becoming AP Bio level. A good high school reference would be Bozeman Science, right? And so this just allows you to get it from a different person, to get a different description, to get a different style, because no 2 teachers teach the same way. And sometimes if one explanation doesn't work, you need to reach out and try to find another. Well, if none of you guys have any questions, I'm going to go ahead and wrap it up. What you should spend today doing is making sure that you've watched 6.1 and 6.2 on structure and replication. Maybe go ahead and take a look at 6.3 on mutation on my YouTube channel. Make sure to finish your vocab and your notes if you can and start studying for Monday's quiz. Any questions? All right. Well, I'm going to go ahead and shut down the Zoom.